We will start with an introductory talk by Luca Murer. He's a fourth year PhD student in the University of Zurich, and he's working on rhinoviruses with professors Urs Greber. And today he will talk about viral epidemics, evolution, and zoonosis. So I'm giving you the power to share your screen.
Very good. Thank you very much, Valentina.
You can start.
Yeah, can you hear me now?
Yes.
Yes.
Okay, perfect. Cool. So thank you very much for the introduction, and thank you very much for giving me the opportunity to speak here on this platform. That's very nice. I appreciate that. And the goal of this is really to give you a broad overview in a very informative and entertaining aspect on general virology, what we're doing, why it is important. So we are following this sort of scheme here, and a side note here, people on the side are doing some gardening work. I can do little about that. You may hear some of this in the background. I apologize upfront. Either way, first I would like to establish a couple of concepts. For instance, the definition of a virus or how hosts and parasites interact with each other, which are going to be necessary to understand the concepts of zoonosis, which I am then going to outline at the hand of two examples in the recent past. So first of all, the term's definition, what is a virus? A virus is an obligatory intracellular parasite. What this means is viruses do not produce their own ATP, they do not have their own metabolism, and they do not produce their own lipids or anything of this sort. They purely rely on host cells to do that for them. Every virus has its own genome, which is either RNA or DNA in nature, and they introduce this genome into a host cell to direct it into synthesizing fresh components to make new virus particles. Which brings me to the third aspect. Virus particles are assembled from scratch every time a virus infects a host cell, and these are then the vehicles of transmission between different hosts. To go closer into the genomes aspect, this is an interesting detail. The smallest genome that we know is of circovirus, that's 1.8 kilobases. Very short, humans have genes that are bigger than that, and Pandora viruses have 2.5 megabases almost, which outlines that we have a wide range of genome sizes in viruses. Their capsids come in all sorts of shapes and forms. We have rod shapes, spherical shapes, bullets, or even the really fancy bacteriophages with these attachments. Some virus particles have envelopes, which are basically lipid membranes that protect their protein shell, which is called a capsid, from recognition by the host immune system. So viruses are very abundant. Actually, they are the most abundant biological entity on this planet. And they are everywhere. They infect every living thing. So every time we eat something, we ingest virus particles. Every time we touch something, we pick up virus particles. And every single time we inhale, we breathe, we inhale virus particles as well. We are in contact with them on a very regular basis, which is not really a big deal because we are resistant to most of them. I'm going to outline this later, however. First, we move on to some host-parasite interactions. When a virus comes into a population, it aims to get established there, and for this it needs to infect other individuals either before it gets purged by the immunity of the initially infected individual or before it kills the initially infected. The rate at which it does so is called R0, or the basic reproduction number. These numbers are different between different viruses. For instance, seasonal epidemic influenza has around 1.5 secondary infections per initially infected person. Measles is very scary, has 15 secondary infections, and the currently circulating SARS coronavirus 2 has between 2 to 2.5 secondary infections, depending on the source and the country that we are looking at. The good news is that we can do something about this in reproduction number. For instance, vaccination in measles has helped very well to take out susceptible people from the population. And unfortunately for SARS coronavirus 2, we do not yet have a vaccine, which is why we have to rely on isolation, social distancing. Of course, for vaccination, it works better the better coverage you have in the population, and therefore it is extremely counterproductive if people start spreading vaccination skepticism, which has become a problem in the latest years. So information of the public is very important in this aspect as well. We are actually infected by a lot of viruses on a daily basis, on a weekly basis, and not all of these cause problems. For instance, varicella zoster, most of the people have seen this virus at some point in their lives, most of us as kids, where it causes chickenpox. This is okay because it doesn't cause any further severe disease or death in humans, which is why both the virus and humans are sort of happy because the virus gets to replicate and we don't get severely sick. So this is what we call a stable interaction between a virus and a host. It also works the other way around. For instance, HIV is a very lethal virus in the long run if left untreated. It is the cause of the biggest pandemic in modern history based on the count of deaths that it caused. This is what we will call an unstable interaction between a host and a virus. Now we can move on to what happens if a virus crosses species barriers, which is what we call zoonosis. As I pointed out in the beginning, we are in contact with many different viruses on a daily basis. And we are living very closely together with different animals because we domesticated them, we keep them as pets, we use them for various reasons, or we just coexist together in the same habitat. They all have their set of stable and unstable interactions with different viruses, and we are living in the middle of this. So of course we get in contact with viruses that are not meant or that are not adapted to infect humans. And what by far the most of the times happens is absolutely nothing, because humans are resistant to viruses that are biologically too far away from their own system. For instance, a bacterium, a virus that normally infects bacteria, would never be able to productively infect a human host cell because it's simply too far away and they're not compatible. A second scenario is when a virus is able to infect humans and cause disease. However, the disease will be so severe that the human dies before it can even pass on effectively the virus to a next individual. This is what we call a dead-end scenario, and a good example for this is, for instance, Ebola virus. This is on individual levels a very scary scenario. However, it is on a global scale not or very unlikely to cause any bigger problems. Which brings me to the third scenario, where a virus particle would infect a human, would cause productive infection and disease. The human then passes it on to a next person, and so on and so forth, causing a chain reaction. Because the virus is not optimally adapted to humans, however, it causes severe disease and even death. And this is the current scenario that we're facing, which is a situation with SARS coronavirus 2. And also this is the scenario that has the biggest potential to cause epidemics or pandemics on this scale. Now that we understand this, we can start looking at how we got ourselves into the situation with HIV. This virus hasn't been in the human population since forever, but only since for about 100 years. And to understand where it comes from, we need to move to Central Africa. There is a close relative that's called Simian Immuno deficiency virus, which is in a stable interaction with monkeys. And this is not usually a problem for humans because the virus spreads by blood-to-blood contact, and this is normally not happening between monkeys and humans. At least this used to be the case until people came up with the idea to hunt and butcher these monkeys for their meat. And it only takes one accidental cut with a butchering knife with monkey blood on it, and you have your interspecies transmission. Roughly at the same time in Central Africa, urbanization became a thing. So the virus now had access to a huge population of people where it could establish itself in the cities. Coming mid-1900s, airplane travel became a thing. So people came from all over the world into these cities from Central Africa, got themselves infected, went back home and spread the disease all over their countries. Because of the nature of the disease, that it takes so long to become noticed, a very long time went by where the virus could have the chance to really establish itself all over the planet until we even started describing it or noticing that something went wrong. And this is the story in a short version of HIV. Now, SARS coronavirus 2, which we are currently dealing with, you may have heard that the virus originally comes from bats. This is very likely to be true. There is a virus isolate that people have sequenced with 96.1% sequence similarity to the human strain. However, there are important differences at the receptor binding site on top of the spike protein here highlighted in red of the virus particle. This receptor binding site serves basically as the key to infect cells, and the key to infect a bat cell does not fit a human host cell, which is why we can be pretty sure that there must have been an intermediate host to transmit the virus from bats into humans. We do not yet know what this species is. However, 18 years ago, if we think back to SARS coronavirus 1, the first epidemic that we were facing, there people identified the intermediate host species to be civet cats. That's how this animal looks like if you haven't seen it before. A similar situation 8 years ago with MERS coronavirus, where camels served as the intermediate hosts between bats and humans. There was a virus isolate found in a very weird animal, which is called a pangolin, that has a 90% sequence similarity between the bat and the human virus. Also, the receptor binding site is compatible with humans, which is why people are currently working with the hypothesis that pangolins were the intermediate host. However, the 90% sequence similarity is not high enough to really explain the role as a direct transmitter, so people are still working on identifying the ultimate virus isolate that was responsible for this. The question, however, remains, how is it even possible that such random animals like civet cats and pangolins transmit their diseases, their viruses into humans? And like for this to happen, we need to get in close contact with these animals, which normally in a natural scenario never happens, unless you dramatically raise the odds of exposure. And that is precisely what people are doing in wildlife markets, for example. So pangolin meat is being served as a delicacy there. People pay a lot of money for this meat. Also, the scales of these animals are thought to have in traditional Chinese medicine healing capabilities or something. So people pay a lot of money for this stuff as well. With civet cats, there was also a very interesting application that people use these as coffee bean fermenters. So they basically feed the civet cat coffee beans, and then they wait for the coffee bean to pass through their digestive tract. Once they poop out the coffee beans, they collect them again and make coffee from this. And this is not a joke, really. That's what's happening, and people pay a lot of money for this civet cat coffee. And as long as there is such monetary incentive to keep these animals so close to us, there will be trade, there will be people like handling them inappropriately. And this is really what gets the species transmissions possible. And even worse, once the species transmission into humans has taken place, the virus now has access to a huge, very densely populated city with people living closely together in poor hygienic standards, where they can just be infected with the virus from where it can then spread all over the planet. So this is what we believe how we got ourselves into the SARS coronavirus mess. And with this, I would already like to wrap up. Point out again that it is never beneficial for a virus to kill or cause severe disease in its host. For this reason, in the long run, it will always either adapt into a stable relationship with the host, or it will disappear from the population. However, this evolutionary process can take a very long time, hundreds if not thousands of years, and we simply do not have time for such a long time to wait for this to happen, which is why the scientific community is working really hard to develop vaccines and drugs that can be used against acute infections. However, taking a step back, it will be extremely important to make people aware of zoonotic diseases being a problem, such that there is pressure on governments to impose regulations on how wildlife has to be handled and used for such applications to reduce the risks of such zoonotic diseases becoming pandemic again in the next years. And with this, I'm at the end already. Thank you very much.
Thank you very much, Luca, for the presentation. So whoever has questions, you can write them in the chat and I can start reading you. So there's the first one. Someone is asking, what is the difference between epidemic and pandemic?
Right. So there are a couple of criteria that the WHO has published on their website. One of them is that it needs to be present on every continent. And also, like, I do not recall every single one of them by heart, but also another criteria is that the WHO actually calls it pandemic. So this has happened in 2008, the last time, where WHO declared a pandemic situation with the swine flu. And actually, they were criticized a lot for that because the flu did not reach such a potential to, like, never infected so many people or killed so many people that this was, in retrospect, necessary. Which is why WHO also has been hesitant to call the coronavirus situation a pandemic until a couple of weeks ago already.
Okay, thank you. So I'll get to the next question. So what about the domesticated normal animals such as cows or pigs? Can they also be intermediate hosts?
Yes, absolutely. So for instance, in influenza virus, pigs do serve as intermediate hosts because they can be infected by bird influenza virus and human influenza virus at the same time. So their receptor glycoproteins are compatible for both of these viruses. So the influenza can recombine in pigs and then pass on the virus into humans. So that's what happened in the swine flu, actually, back in 2008, 2009.
Okay. And the next question is about vaccines. So what is the good process to create a vaccine and what is the time scale to do it?
Right. Yeah, that's a very good question, actually. So normally, people take different approaches to make vaccines. Depending on which virus you are dealing with, you have different possibilities to do so. So for instance, you can take a live virus and somewhat inactivate it, make it weaker, such that it cannot really cause any disease anymore, but your immune system will still recognize it and trigger an immune answer, which will then protect you from the real virus. There are a ton of other approaches on how this is possible, and people are now working on several of them at the same time. One thing I would like to point out here is that the development of a vaccine, including clinical trials, including upscaling production to large scale, normally takes about 10 years. And people are now trying to accomplish this entire process within 1 year. So this is a tremendous effort that people are doing here. And if they really manage to get this done, then that would be like really a cool thing. So yeah, let's hope for the best here.
Okay, so next question. So then I will read those that are here and then I will stop asking questions so that we go to the next talk. So someone asking if plants can also be source of interaction and also cure animals could be a useful and realistic strategy to prevent epidemic on humans.
Yeah, plants, I think this is probably theoretically possible. However, I do not like I'm not aware of an example on where this has happened really, but I think plants are biologically already quite far away from mammals or from humans. So I think this is rather unlikely, but I would not exclude it. And the second question, curing animals. Yes, I think this would be feasible. However, there is way too much going on in terms of there are way too many viruses in every single different species, and we cannot even keep up with the human viruses that infect us. So unless there is like an important economic need to deal with these animal viruses, it is probably economically more interesting to focus on the human viruses.
Thank you very much, Luca. Thank you. I would have a few more questions, but maybe I will ask them at the very end if you're all still here. So what was the difference in the between the SARS-CoV-1 and this second one? Why was the number one limited in space and time and this one spread all around?
Right. Yeah, that's a very good and interesting question. Actually, we are not at this point perfectly sure about the reasons as to why this is the case. A couple of hypotheses involve that there is a furin cleavage site present in the SARS coronavirus 2 that enhances the spreading ability within humans. And so this probably very likely plays a role for the ease or the rate at which the virus spreads. But there are other factors that play a role here in this aspect, such as, for instance, the international measure of connectivity between the country of China and like any other country at this point. So 18 years ago, this was a lot lower. There has been a lot less airplane traffic and generally people traveling from and to China. So this for sure as well played a role in the ease at which the virus spread all over the planet.
Okay, then I got to a question. I don't know if the person who asked is still there, but does SARS-CoV-2 coronavirus 2 use some kind of integrase?
So here I'm walking on thin ice because I am not super familiar with the biology of the virus at this point. However, since SARS coronavirus 2 is not a retrovirus of any sort, so the genome is actually RNA nature, it has not been described to integrate its genome into any other host genome at this point. However, how this translates into whether an integrase is being present on the viral genome or not, this I am not completely aware of.